Hello and welcome to our next episode of VCE Biology with me, Mr. Dean. Today we'll be heading somewhere over the membrane. Moving on from our previous topic in our last episode about the plasma membrane, we're going to be talking about what kinds of things are actually able to cross the cell's plasma membrane and how they might do so. We're going to begin with a bit of a recap of the structures of the plasma membrane that we looked for in our previous episode, and then we'll be talking about how molecules are actually able to cross the plasma membrane and how things are actually able to move across in different functions and ways. So to begin our recap of the plasma membrane so far, we started off by saying that the plasma membrane exists within this region between the extracellular fluid that was on the outside of the cell, and then we had our intracellular fluid that was within the inside of the cell that include the cytoplasmic fluid. Structurally, we began by talking about our phospholipid bilayer that was over here, that we said was composed of two key parts, and that was included our phosphate heads and our lipid tails. Our phosphate heads that we just said were polar, and then we had these lipid portions that we said were non-polar. And so we said that this important part of the phospholipid bilayer gave the plasma membrane an important way in which to both repel water and attract water. So we said these phosphate heads, as they were polar, they were our hydrophilic, and they were actually able to attract water. And in doing so, they attracted the surface of liquid across the plasma membrane. The inside part, when we're talking about the lipid tails, these were non-polar and they were said to be hydrophobic, which means they were water repellent, they don't dissolve in water, and they created this barrier that prevents water from just being able to seep within the cell, that gave us actually a little bit of structural sort of conformity and gives us a little bit of structural integrity to the cell as well. Our next structural component of the plasma membrane we talked about were these cholesterol components here. These cholesterol components, we talked about them being lipid molecules that sort of sat within the plasma membrane. We said as the lipids they were non-polar, they didn't dissolve in water, and they added an important aspect and characteristics of the plasma membrane, that they were fluid during temperature changes. Just going to put a note here that they were fluid. And during warmer temperatures, we said that these cholesterol components, they were actually able to prevent the plasma membrane from simply falling apart, and during really cold sort of moments, they prevented the plasma membrane from sort of closing in together and pushing together and crystallizing so that the cell didn't as easily freeze. Our next components of the plasma membrane we talked about included these proteins here. We started off by saying that we had two different groups of proteins. We started with our integral proteins, which this might be an example of, for example, and these were our proteins that could be found permanently fixed within the membrane. This protein I've just drawn an arrow through as well could also be considered to be a transmembrane protein. That was said to be a protein that actually spanned both sides of the plasma membrane, but we also made the distinction that we might have integral proteins that are only on one side or perhaps either side of the membrane. These integral proteins might have a variety of functions. For example, they might act as pores, allowing things to be able to travel straight through them, or they might actually perhaps act as an enzyme on one side, or they might have potentially a receptor on another side, or a variety of other functions. Our next family of proteins we also talked about included these peripheral proteins down here. These peripheral proteins, which I don't think quite are featured on this diagram here, so they included these proteins that sort of didn't exactly anchor to the plasma membrane themselves, but they sort of sat maybe outside the protein, they sat outside the plasma membrane, or they sat on top. We said that these peripheral proteins were really important in the similar sort of functions, acting as enzymes, or perhaps acting as transport proteins, or perhaps even acting as pores within the membrane. A defining characteristic of these peripheral proteins as opposed to their integral counterparts were that they were not permanent fixtures within the plasma membrane. They sort of were able to actually move around, and they weren't always found within the plasma membrane at any given time. Our final components of the plasma membrane we talked about included these glycoproteins here and subsequent glycolipids that I don't think again are quite featured on our little structures here, but our glycolipids, very similar to our glycoproteins, they were made of one part of a simple sugar and then attached to either a protein or a glycolipid. These glycolipids or glycoproteins could be attached to any sort of protein featuring. That could be a peripheral or an integral protein. These carbohydrate chains then may be attached to any of our lipids that feature in the membrane, And then they would just be composed of chains of our simple carbohydrates or simple sugars that we've talked about. These glycolipids and glycoproteins were really important for cell-to-cell adhesion. They helped with some cell-to-cell signaling, and they were also really important for being able for other molecules to be able to attach to the cell, including our hormones and our antibodies. Our final part we covered in our previous episode about the plasma membrane was the conception that our plasma membrane followed this sort of model that we talked about that was called the fluid mosaic model. The fluid mosaic model, we said, described the plasma membrane as having both a fluid component and then a mosaic component. So this fluid component or characteristic, we said that the plasma membrane was a very dynamic structure. It didn't really stay exactly how it was. It was sort of very loose. It was sort of very fluid and dynamic. It kept on moving around. It sort of swelled as it sort of floated around in the liquid and fluid that it surrounded it. The mosaic component described how there were many different parts of the plasma membrane, as we've sort of detailed in this little recap. It had many different parts, including the polar and non-polar components of the phospholipid bilayer. It had embedded proteins and lipids and carbohydrates, and it really formed this structural mosaic. Now moving on to our membrane movement portion of this week's video, we're going to be talking about how molecules actually move across the plasma membrane. So many different types of molecules are actually able to move across the plasma membrane into or either out of the cell. Just take a moment here to answer the question that I've got here on this slide, asking what kind of molecules do you think are actually able to easily cross through the plasma membrane? Here of the selection we've got water, ions including say potassium or sodium, oxygen as being O2, carbon dioxide, monosaccharides such as glucose, and lipids. Okay, taking some time now to look at it, hopefully you guys will have remembered from our previous session, we talked about the plasma membrane having our insoluble double lipid structure within it that prevents molecules that are easily dissolvable in water from passing through. So obviously we've got water, ions, and things like monosaccharides that can't actually easily pass through the membrane because they naturally sort of dissolve in water. Our carbon dioxide, our oxygen, and our lipids, which are usually insoluble, and when we talk about them, they're also non-polar, they're actually able to sort of pass through the plasma membrane. Here we've got a diagram representing how different sorts of molecules actually are able to pass through the plasma membrane and for what sort of reasons. So here we've got our water that's able to pass in and out. We've got some oxygen that's able to pass in, usually for the reason of respiration, where we can get some energy. We've got carbon dioxide exiting, usually as a product of respiration, because it's usually a byproduct of respiration. We've got some nitrogenous waste here, various ions such as potassium or calcium, our monosaccharides such as glucose that we use for energy, some of our lipids that we may also use for energy, or some potentially structural components, and amino acids, which are the building blocks of proteins. In talking about the movement of molecules across the plasma membrane, it's really important to consider the factors that affect how these molecules were actually able to pass through the plasma membrane. Here I've got a list of them that includes size, charge and polarity, and whether or not the phospholipid bilayer is permeable to the substance. So it's really important to consider that larger molecules are going to be really difficult to get through the plasma membrane. Charge also affects how a molecule is able to pass through the plasma membrane. Polarity, including if a substance is either polar or not polar, and whether or not the phospholipid bilayer is actually permeable to it. I've got this diagram here on the right trying to be sort of synonymous to how these membrane sort of actually operate with molecules trying to pass through them. Here you can see different sort of shapes that I think sort of represent how these molecules are sort of permeable. So you can see that there might be, say, specific protein channels that allow certain types of molecules to actually go in. So if the shape of the molecule or potentially the size doesn't match these particular pores or doesn't naturally just sort of fit in because it's too large, it may not actually be able to pass through the membrane. For the next portion of the video, we're going to be talking about the actual processes that go along and force the molecules either into or out of the cell across the plasma membrane. The first of these is we're going to be focusing are what we're going to call our passive processes. These passive processes, as I've said in this first dot point, refer to ones that sort of naturally happen and require no energy. So you can think of these as requiring no energy and sort of being a natural tendency. They just sort of occur because they don't require any extra energy. They just sort of happen within the cell. So our passive transports refer to the fact that no energy is expended to move molecules across the membrane. And the first one we're going to be looking at here is diffusion. As I've said in the second dot point, diffusion is the natural tendency of particles to move from a region of a high concentration to a region of low concentration. I've gotten the second dot point just below it. Consider how ink or food dye spreads in a solutes in water, the solvent. So consider if I'm putting a drop of food dye in water, it sort of naturally spreads out. And we'll look at this in our next slide as well. Diffusion occurs both in liquids and gases, and we describe this movement of molecules from the region to another region as the molecules traveling down their concentration gradient. So when we're thinking of how molecules travel through the plasma membrane via diffusion, we're thinking of molecules such as oxygen, carbon dioxide, and certain alcohols here such as ethanol and methanol. Here I've got a sort of visual representation of what I was trying to describe earlier, being that we've put these few drops here of the food dye or potential ink, and these molecules move down their concentration gradient, being highly concentrated here and very highly concentrated here, and moving out to regions where there's a very low concentration, and this is by the process that we've described up here, being diffusion. As you can see in this final sort of jar, the ink or the food dye has really moved out to these sort of positions where the ink is of a lower concentration. Looking here at another visual representation of how diffusion works as a passive process, you can see that I've got these two sort of beakers, and we've put this first concentration, perhaps these are blood cells, perhaps this is food dye, and I've got this first region, and I'll label this region 1. And as you can see in region 1, we've got a really high concentration of the red particles just in this little zone. And so if I describe this in a sort of graph, and on the y-axis of this graph, I've sort of got my concentration, and I've got my regions down here. I can see that in this region 1, I've got a really high concentration up here, and there's a natural tendency for these particles to want to spread out and go to sort of this second region where the concentration of the particles is lower. When we're talking about concentration, it's important to consider how many molecules are in a given space, and as you can see, there's lots of molecules in this small given space here, and so the concentration is really high. And as you can see in this second visual representation of the beaker, in the same sort of amount of space as I've got in my first region, there's not as many particles in this region, and so we've got a lower concentration. And this describes diffusion being that sort of natural tendency for molecules to want to spread out and move from a region of a high concentration to a region of a low concentration. Moving on to our next passage through the membrane, we're going to be talking about here osmosis. As I've got in my first dot point, osmosis is very similar to diffusion, but it involves a semi-permeable membrane, also sometimes labeled a partially permeable membrane or a differentially permeable membrane if you see them within exam questions. These membranes are usually defined by the fact that they allow water to be able to cross them, but block other solutes from crossing them. So for example, I might be able to allow water through them, but certain other ions such as say potassium or sodium, or perhaps larger solutes such as glucose or other things like that, aren't actually able to cross this membrane. We can say that this is actually similar to the plasma membrane in that the plasma membrane is actually easily able to allow water to pass through the aquaporins or pores that we talked about earlier. I've got a question here on the bottom of this slide asking if I have a container with two sections and it is separated by a semi-permeable membrane and a solute such as sugar is added to one side of the container, what do you think will happen and why? Perhaps consider using some of the knowledge that you've gained thus far thinking about how molecules may actually naturally move in a sort of diffusion-esque manner. Hopefully you guys would have been able to get there. So just as like diffusion, osmosis will occur and the water will travel from an area of a high concentration to an area of a low concentration. And when we're thinking about that, I'd like you to start thinking about that the fact that the water will move from an area where it's in an abundant or lots of free water molecules are. So for example, if I'm looking at the side where the sugar was added, you can see that there's actually lots of free water molecules here. I've got lots of these free water molecules. So it's in this region of a really high concentration of free water molecules. And then over here, I've got this region where there's not as many water molecules. And so there's the natural osmosis tendency, very similar to diffusion, where I'm going to go from this region of a high concentration to this region of a low concentration. And we can see that in the second diagram drawn here, where the water has all gone to this side, and it's pushed the water up higher here in the beaker. Similar to the sugar molecules on both sides of the permeable membrane, you can see that the sugar molecules actually haven't changed. But if I look at any given section, say this little section here, and a little square here, I can see that the amount of sugar and the amount of water in a given section is roughly the same. So just to recap that, we put sugar into one side of the semi-permeable membrane, water moved from a region of lots of water molecules to a region where there was very few water molecules in comparison. And we can see that here, where along a semi-permeable membrane, the water is actually pushed right above the separated section. So just recapping here of the passive process of osmosis and what we saw on the previous slide, the net movement of the water molecules from the diluted region to the solute concentrated region demonstrates water moving down its own concentration gradient. And we call this an osmotic gradient because when we're talking about osmosis, we're thinking about the movement of water. Typically also we're talking about the pressure that moves these water molecules across a semi-permeable membrane, and we call that the osmotic pressure. So again, just to recap that first dot point, we've got the net movement of water molecules from a region of a high concentration or a high osmotic pressure to a region of a low osmotic pressure. We call this the osmotic gradient. Just to finish up on osmosis, I'd like to give a little bit of a definition here or two. So osmosis is typically defined as the net movement of water along its osmotic gradient from a region of a high concentration of free water to a region of a low concentration of free water across a semi-permeable membrane. You'll see that I've underlined the words free water here first. Free water is typically just defined as the free water molecules that surround a solution that actually aren't sort of interacting with a solute like sugar. So typically when we put sugar into a region of the say a beaker separated by a semi-permeable membrane, some water will just naturally go around that sugar, and then we'll also have free molecules that aren't interacting with the sugar. And so when we're talking about free water, we're talking about the water molecules that aren't interacting with the sugar molecule. And again, just to reiterate, we're always talking about a semi-permeable membrane when we're talking about osmosis, and being that a membrane that actually allows water to move past, but not other solutes. Perhaps a more simple definition I've got down here in a second dot point is osmosis describes the movement of water down its osmotic gradient to a region of a low osmotic pressure across the semi-permeable membrane. Moving on to our next passive process, we've got what I've called facilitated diffusion. Facilitated diffusion is actually again very similar to diffusion, but involves the movement of molecules through transmembrane protein channels. So we saw this when we were talking about our integral proteins, and in particular our transmembrane proteins. These movement of these molecules move again down their concentration gradient via specific protein channels, likely transmembrane proteins. These molecules don't normally pass through the plasma membrane and thus need proteins to help facilitate their transport across the protein. This can be either into or out of the cell. This process is specific to the solute itself and usually occurs faster than simple diffusion. When we're talking about simple diffusion, we're really just talking about the broad concept of diffusion as molecules move down their concentration gradient. So just to sum up so far, facilitated diffusion usually involves potentially ions, or potentially say amino acids that don't normally pass through the plasma membrane, and so they need help, potentially through these transmembrane proteins that facilitate their movement. Here I've got a little diagram showing the differences between facilitated diffusion and normal diffusion. So over here we've got diffusion, so this potentially might be a CO2 molecule, an oxygen molecule, or potentially an alcohol that's just able to naturally sort of move through the plasma membrane. Up here I've got two different examples. These green triangles perhaps maybe say ions such as potassium or sodium, and they require this transmembrane protein to actually get through the cell. And again, they're moving from a region of a really high concentration here out of the cell into a region that's a really low concentration. I've got a second part of this diagram here simply showing another kind of transmembrane protein. The only difference between these two is that this one seems to show that there's actually some sort of conformational change. So you see this little protein channel here. It looks like it might actually have to move in order to be able to pump these sort of molecules down their concentration gradient. But just to reiterate, facilitated diffusion again is a passive process, and so it actually doesn't require any energy to occur, as all of these molecules are moving towards their natural tendency from a region of a high concentration to a region of a low concentration. Moving on away from passive processes, we're going to now be talking about active transport. So active transport, as opposed to passive transport, involves the movements of molecules from a region, conversely, of a low concentration to a region of a high concentration. So we're actually moving things against their concentration gradient when we're talking about active transport. The other difference between passive processes and active transport is that active transport actually requires energy. So typically when we're talking about active transport, we're talking about the expenditure of ATP. And just to reiterate, instead of things moving down their concentration gradient, we're actually moving them up. So thinking about the energy that you might need to expend, say having to walk up a hill. So if you're walking up a hill, you're starting at the bottom of the hill and you need a lot of energy to get up the hill. So active transport requires that energy, as opposed to our passive transport where you might actually just need to normally roll down a hill and you don't need to actually expend any energy. Typically when we're talking about active transport, we're talking about proteins being involved to pump these, as it's not a natural process. So I've got in my last dot point here, why do you think we may need to use some active transport? And I'll just let you have a moment here to pause and have a think about why we actually might need to expend energy to pump molecules such as proteins or solutes or ions into the cell. Hopefully you came up with a great answer there. I've got another slide here on active transport, just trying to show it to you. So here we've got some different examples of some molecules that might actually need to be pumped into the cell, and we've got an example of what this protein that facilitates active transport might look like. An important part of this diagram here is the expenditure of ATP as cellular energy that enables us for this protein to be able to move things from a region of a low concentration to a region where they're already in a high concentration up here, against their natural tendency. And this is at the expenditure of some amount of ATP through some sort of transmembrane protein here. The examples of molecules that travel across the membrane via active transport include things like glucose, sodium, potassium, and proteins. Glucose, as you can imagine, is always really needed in the cell, so we may need to expend energy to make sure we get it, especially within the epithelial cells of the gut, as it's a form of energy, and cells really always need a form of energy to be able to conduct their cellular processes. Sodium and potassium are also really important ions in keeping a balance in our cytosol and in our sort of intracellular fluid to make sure that our normal reactions can occur. And proteins are also a really important part of what we make in the cell and what we export and what we need within the cell. Just to make sure we're clear, when we're talking about active transport, we're also talking about things being able to go into the cell and things that are able to go out. So sometimes we'll expend energy making sure we get in things, so potentially glucose in our extracellular fluid, we want that in our cells as an energy source, so we'll make sure we bring that in. But in the case of, say, proteins, sometimes we'll make proteins within our cell and we'll need to expend energy getting them out. Our final slide here looks at some of the different processes that sort of exist under the umbrella of active transport. Our first example here is exocytosis, a form of active transport that involves transporting materials across the membrane out of the cell in membrane-bound vesicles. So if you remember earlier, this might involve some of our proteins being made in the cell, and sometimes I'll wrap them in a membrane and sort of push them out of the cell. This would be really important when a cell is making a particular hormone protein or perhaps an enzyme, and it needs to export it to the rest of the body, so it moves it out of the cell via exocytosis. Our next example is endocytosis here, which is a form of active transport that involves transporting materials across the membrane and into the cell in membrane-bound vesicles. This might be where a cell engulfs potentially another particle or where it wants to engulf some fluid. Finally, I've got two more processes down here being pinocytosis and phagocytosis. So pinocytosis is essentially the endocytosis, but involves taking in extracellular fluid to the form of vacuoles. This might be the case when we've got some ions or some important fluid that a particular cell might want, and so it brings it into the cell in the form of a little vacuole. Phagocytosis is essentially endocytosis again, but involves the cell engulfing large particles. So for example, this might be the case where if we've got, say, a white blood cell or something, it might engulf some large particles that are potentially harmful to the body, and it might engulf them so it can destroy them. And that's all for this week's episode of VCE Biology. I've been Mr. Dean. I hope you've enjoyed. Bye-bye.